Genetics for today. Talking about genetics for today. To start off this new unit, we're going to learn about somebody called Gregor Mendel. You see his picture over there on the right-hand side of the screen. Mendel actually showed that traits are inherited as alleles from our parents. That's what he means by discrete units. And you might notice, okay, one good observation, I can't remember by who in third period, but very good observation of, hey, this person looks very religious. Well, that is because Gregor Mendel was actually a religious man. He was an Austrian monk. Very intelligent human being, did a lot with genetics. He's also nicknamed the father of genetics because he laid out all of the groundwork that was necessary to understand genetics in the way that we understand it today. Obviously, there's plenty of other people that have worked on genetics since him, but he's the one that laid out the basic groundwork that we needed for what we know today. So traits are distinguishing characteristics that are inherited. So traits like, okay, your hair color, or your, I don't know, the rate that your fingernails grow. That's all based off of genetics. Genetics is the study of biological inheritance patterns and variation. As you know, as I mentioned already, Gregor Mendel, he was the one that determined all of this. And a lot of people in his day thought that, okay, you end up getting like an even split of, okay, you have 50% of mom's traits and 50% of dad's traits. Well, it's true that you get 50 and 50 of their genetic material, but on the trait side of things, you might not necessarily express the traits that they did. So there's a chance that you might get traits that were expressed 1 or 2 generations ago. So either traits from your grandma or your great-grandmother. There's a chance of that. Or same thing for the fellows, your grandfather or your great-grandfather. It just really depends with genetics. It's really cool that things like that can happen. For example, another thing that is determined by genetics is determination on a higher chance of getting twins or triplets in a family. So for example, with me, I'm actually, my generation is due, in my family, my generation is due for twins. So I would be due for twins if I were to have kids. Now, it's obviously not a for sure thing. Like, nothing in genetics is a for sure thing. It's all probability. A lot of the probability that we do today in the first couple of days of this unit are going to be fairly simple probability. So not a lot of overly intense mathematical stuff for this unit, but it's always really cool to look at the different probabilities that we have to be able to inherit specific traits or specific characteristics from our parents. Okay, you need everything on this slide except for the blue. So Mendel did 2 different experiments, all on pea plants, by the way. It's very interesting because he did one on the flower color, which was super duper basic. That's the one we'll touch on today. And then he did a lot more characteristic building things and figuring out some things with characteristics through some other experiments as well. So some of the key decisions he made in his experiment, he used purebred plants to start. So I'll explain what purebred plants are here in a little while. And then he had control over breeding. Now, how do plants breed? Do they breed sexually or asexually? Asexually, because they are pollinated by pollinators. And what's an example of a pollinator? What would be a bee? The common thing that we think of is a bee. They are able to carry the pollen. That's how we breed plants. So he was able to crossbreed and actually selectively go in and breed certain plants with certain plants to get specific characteristics or specific traits that were advantageous to production of peas. He observed 7 different traits, 7 different either or traits when he went through and did his pea plant experiment, whether it be a long pea pod or a short pea pod, the color of pea, the color of flowers on the peas, if the peas are wrinkled or not, if they're yellow or green. There's a lot of different characteristics that he went through as he was going through and crossbreeding these plants. Definitely interesting, definitely a change of pace for science, because a lot of times when we go through and we do experiments, we always only focus on one variable. As for genetics, you can kind of piece it together to where you can have as many traits as you want to focus on. It's just kind of hard to keep track of. Today we're going to do some simple Punnett squares, but what he did is he had 7 different traits that he had to focus on. And it really does kind of get messy when you're focusing on 7 different traits. So it's kind of crazy to think that, hey, you can track all of that. But it's definitely easier to keep track of either 1 or 2 traits, not 7 of them all at one time. Okay, it looks like we are all good. You only need the top bullet point on this one, please. Top bullet point. On the bottom is showing the process that he went through to be able to selectively pollinate these different plants, or selectively crossbreed them. So when he went through, he went and cut off the male part of the flower. And yes, there is a male and female part of the flower. First, he cut off the stamen of the flower. So the male part of the flower is called the stamen, and the other part, the female part, is what he actually brushed with his little brush, kind of in place of pollinators. So he didn't use bees or anything, because that could throw off the genetics and could be getting pollen from other plants. So he personally went through and fertilized all of these plants himself. He went through and fertilized with this little brush, kind of does the same thing as a bee's legs because it traps pollen on it, and rubbed it on the pistil, or the female part of the plant, and boom, he was able to crossbreed the plants that he wanted. Yeah. So he went through and he was able to breed these pea plants in a way that he found conducive for what he was particularly looking for, or particularly studying. Okay, main point on this one that I need you to do is I need you to draw the bottom part and have the words as well. If you don't want to sit and draw flowers, here's what I would do. I would just put with 2 circles, and you're going to go, okay, this one's purple, this one's white. This is in our pea generation. Those were crossing, and they ended up making our first generation. That's what it means by F1. So F1, and we're going to have purple and purple. And then we ended up breeding those 2, and there's 4 offspring here. 1, 2, 3, 4. And you're going to have purple, 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 and white. And we're going to show that here in Punnett squares here in just a second. But that is a simple way of drawing it. So if you want to draw that bottom picture, I would just draw it with letters instead of going through and doing it that way. So you can go through, draw that bottom part. Make sure, like I said, that you include these portions by the arrows. So those, all 3 of them that are on there. And then once you're done, we will go with the longest part of our notes for today, which is going to be making those Punnett squares. So again, this is all going to be recapped when we go through our Punnett squares. This is kind of a different way of showing, that. All right, okay. So here is kind of the nitty-gritty details of how he kind of went through and looked at traits. We're going to focus mainly on color first. In the coming days, we're actually going to be able to do several different traits when we go through and do our Punnett squares. So first, we need a little bit of information for our Punnett squares. So I'm going to provide that to you because he crossbred two purebred, not pure bed, purebred flowers, two of them to be exact, two purebred flowers. We are struggling today. We are struggling. Purebred. So two purebred flowers. You have a purple and you have the white. Because this is going back to that generation of flower that was all the way on the left side of the screen, that very first one. We bred a purple purebred and a white purebred. Okay, two terms that we're going to continue to use pretty frequently, especially in this unit, is genotype and phenotype. So we need to go through and identify each for each of these two flowers before we can do our crossing in our Punnett square. So genotype and phenotype. And can anybody tell me which one of these two is the one that is the actual visible expressed trait? Is it phenotype or genotype? Phenotype. It is the phenotype. Yes, it is phenotype. So phenotype is the actual expressed trait. And what is the trait that we are looking for? What is the trait that we are talking about? Yes, color. Yes, color. So purple for our first phenotype and white for our second phenotype. Then genotype, I know we talked about genotype before, but it's going to be a little bit different for this one, especially when we get into our Punnett squares, is going to be letters. You're going to just see a whole bunch of letters. So, for example, when we go through and we do genotype for our purebred purple, it's going to be dominant because purple is a dominant color. White is actually a recessive color. So we have a purple genotype of two big letter P's. It's very, very important that you have big letter P's because capitals and lower cases do matter when it talks about genetics. Okay, then we have our second parent, which is going to be our purebred white, and they are going to be two little letter P's. Two little letter P's. That's what we're going to have for our genotype. So you have our genotype and our phenotype. Some new terminology that we also need to know, aside from genotype and phenotype, is we need to have a word or two words to be able to describe our genotype. So if you look, they have the same letters, and they are both capital for our first parent. So I told you already that it is dominant because big letters are dominant, so we can at least have that part. But there's a word that comes before dominant, and I'm just going to do dom for short. There's a word that comes before dominant that means the same. There's an H word in science that we use to describe something that is the same. Homo. Homo is the prefix for it, so this is going to be homozygous dominant is our genotype up top. So homozygous. So homozygous, this is supposed to be an O and a U. Homozygous dominant. So it's homozygous dominant for our top parent. Now, what do you think about our bottom one? Are they the same letters? Are they the same case size? Are they both lowercase?
Yes.
Yes. So it's going to be homozygous, and are they dominant or recessive? If the top one's dominant, then that must mean that the bottom is recessive because they're little letters. So we have homozygous recessive. I'm just going to do homo for short. Homo and recessive. Okay, we have that. Homo recessive and homo dominant is the two that we are crossing for this particular instance, this particular breeding. Okay, what we are going to do is we are going to do what is called a mono hybrid cross. So a mono hybrid cross. And tomorrow we are actually going to do a dihybrid cross. If you remember the prefix mono, what does mono mean? So when somebody says mono e mono, it means one on one. So mono is one. So we are looking at one trait. One trait. And the trait we're looking at is color. Color. Looking at color. Okay. Now we get to do the fancy stuff. We get to actually do our Punnett square. So you're going to draw a box that kind of looks like a cartoon window of sorts. It's going to be a box, and you're going to separate it into 4 little boxes. 4 little boxes is what we are going to need on that. So you need to draw something like that. That's how we do our Punnett squares. Okay, so when it comes to our Punnett squares, we always have parent 1 up top. So parent 1, and parent 1 has big P and big P. So big P right here and big P right here. When we do dihybrid crossing, there's going to be 2 letters above each of those spots. And I'll explain that when we get to it tomorrow. Then, on the left side, if we have parent 1 on top, then that must mean that, hey, parent 2 is going to have to go over here on the left-hand side. So parent 2, and we have little p and little p. Okay, now we are ready to actually do the hard work. We go through, and we take 1 letter from mom, or 1 letter from dad, whichever parent first, and you come in here and you do both. So we have a big P and a little p. We get this because we drop down the big P from parent 1, and we get little p from parent 2, and boom, you have big P, little p. Okay, what should our top right box be? What should our top right box be?
Green P, and then blue P.
Yes. And to elaborate on the green and blue, green is actually going to be our dominant big P, and our blue is our little p. So big P, little p. Okay. What should box 3 be? Should it be the same?
Yeah.
Yes, it should be. I would agree. And same thing with box 4. It's going to be the same. So we have big P, little p, in both of those boxes. Okay, so now we got the hard part out of the way. Now we have to go through and determine a couple of things. Okay, if something has one dominant letter and one recessive letter, are we going to express the dominant trait?
Oh, yeah.
Nod your head yes. Nod your head yes.
I'm doing it sideways.
You're just seeing if I saw. That's what you're doing. Okay, so since we have our dominant big P in all of these boxes, all of these flowers are going to be purple because purple is our dominant color. So all four of those boxes are going to be purple. That's what those are going to be coding for. So 4 purple flowers. Okay, now we have to go through and determine our genotype ratio and our phenotype ratio. So if we were going to start off with our phenotype ratio, because that one's going to be the easiest. Phenotype ratio. All right, what is our phenotype ratio going to be for this particular instance? What's our phenotype ratio going to be for this particular instance? How many purples do we have to how many whites?
4 to 0.
4 to 0, so we have all purple. All purple. Okay. Now, we have our genotype ratio. Geno ratio. All right, looking at our box, what do we have using our new terminology that we learned up here between homozygous and heterozygous? What do we have here if we were describing this? Homo means the same, hetero means opposite, by the way. So if we look at this, are these two letters the same? They are not. So that must mean they are what? So it's hetero. Heterozygous. So we have all hetero. And are these dominant or recessive? You guys have already answered that question, so you should know. Are these all dominant? Because are they all expressing a dominant trait? They're dominant, yes. All heterodominant. Okay, boom, there you go. Congratulations, you just did your first monohybrid cross, your first Punnett square. I don't want to scare you, but that'll be the easiest one that you do. That'll be the easiest one that you do. So it should be pretty simple as long as you can keep track of your letters. So monohybrid crossing is actually the most simple one. In college, I think they had us do trihybrid crossing, and there was another one after that. So where we were actually following through with 4 traits. It seems like a lot, but if you just track your letters and figure out, okay, how many are actually being bred with how many, you're able to do Punnett squares. I think that is one of my favorite things in biology out of all things is our Punnett squares. Okay, does everybody have this or has a picture of this? Okay. Perfect. Good, good, good. All right. Okay. So we're going to move on now because we are going to breed those 2 plants that we just made. So we are going to have, what I mean by that is we are going to have big P, little p, crossing with big P, little p. Now, if we go through the same process here and we determine, okay, genotype and phenotype, we should know all this because we already just wrote that. So geno and pheno. What is our genotype that's being expressed here? What are the letters? Both. Yes, they're peas. But
Oh, purple and white. Purple.
no, that's a phenotype. You got the phenotype here, so you got at least that. The hemoja. I'm not understanding the hemoja. You guys are making it too difficult on yourselves. It is going to be our two letters that we have up here. We got a big P, and we got a little p. Got a big P, little p. But the word that you're trying to come up with when you're like humana is the heterozygous, because they're not the same. That's why they're not. Heterozygous dominant, because you still have the big P there. Okay, we got all the important factors out of the way. Now we can actually go through and do our Punnett squares. So let's go ahead and draw our nice little window, our window to see into genetics here. The window into life. Here we go. Okay, we have, I'm going to keep this the way that it is here. We are going to have big P and little p. Right, we're going to have P there and P there. P and P. All right, so we have parent 1 and parent 2 on there. Now we need to go through and look at our box and determine what we're going to have in each box. So if I'm looking at our top left box, our box number 1, what should I have for my two letters?
Both green peas.
Yes, but are they big P's or little P's? They're big P's. See, the reason why I'm such a stickler on being able to say big or small is because it really does matter. Because if you have a big P somewhere in any of those, they are going to be dominant. Versus if they're all little p's, they're going to be recessive. So whenever I say what are the letters, I need, hey, big P, big P. Color coding just helps to visualize what parent they're coming from. Okay, so then we have box 2 on the top right. What is box 2 going to be?
Big P and little p.
Yes, thank you, Emma. Big P, little p. Big P, little p. Big P, little p. All right. Now, bottom left box, what is it going to be? Big P and little p. I would agree. I would agree. That's a very great observation. Big P, little p. All right. Not a trick question here. What is box number 4 going to be? Yes, Emily. Little p and little p. Yes. Little p and little p. Little p and little p. All right. Okay. Now we got all of that out of the way. Now we need to determine what our phenotype is going to be. So if we have dominant and dominant, what color is it going to be?
Purple.
Yes. It's going to be purple. Going to be purple. Going to be purple. All right. Now, if you have big P, little p, what are we going to have? It's going to... I got you guys. You said white. It's purple. It's purple. Now, why is it purple? Why is it purple? Anybody tell me? Because there is a dominant gene there. Yes. Right here, you got the big P. That is why that is going to express purple. So we have the same situation going on down here in this bottom left box. Okay, there we go. All right, we have one box. Box number 4, what do we think the color is going to be? White, yes. Because there's no dominant in box 4. So this should be white. Okay, we are done with our Punnett square side. Now we need to determine our ratios. We need to go through and determine our ratios. This one's going to be a little more difficult. We'll start with phenotype ratio first because that's the easy one. We'll get the easy one out of the way. We'll end on the hard one for today. Phenotype ratio. All right, phenotype ratio. Okay, what's our purple to white ratio?
3 to 1.
3 to 1, 3 purple. So 3P to 1W. 3P to 1W. All right. Now for the difficult one. We'll see if you remember terminology on this here. This will be a good little test here. Good little test. Genotype ratio. Okay. If we have two of the same letters, what is the terminology for it? Two of the same letters.
Oh, that's dominant heterozygous.
I heard homo over here. I don't know who said homo. Homozygous, yes. And if they're big letters, they are what? They're Yes, thank you, Lucy. Dominant. So, talking about box 1 up here, box 1, we have 1 homozygous, 1 homodom. 1 homodom because we have dominance. All right. Now box 2 and box 3. What are we going to have for box 2 and box 3? What's our terminology there to describe what's going on? It's heterozygous. Lucy's right. Now, is it dominant or recessive? It is dominant. And why is it dominant? Because there's a big capital P there is why it's dominant. Big capital P right here and right here. So we have 2 hetero. So hetero. 2 heterodom. Okay. We got one last one because we've accounted for 3. We have one more to account for. What is the terminology for the bottom one? They're both the same letters, so what should the first word be? Homozygous. And it's recessive, as Lucy was saying. So homozygous recessive. 1 homorec. 1 homorec. 1 homorec. Okay. And just like that, we're done. We're done with all of the Punnett square stuff for today. The easy Punnett squares. You now can successfully go and do monohybrid crossing on anything that you want to do. As simple as that. That covers pretty much all the details that you'll need whenever you do monohybrid crossing on assignments or on the test. Of course, like I said, we're going to do dihybrid and trihybrid later on, which is a lot more complex. But for now that we have the basics, we kind of know how that's going to work. Okay, everybody has this drawn, correct? Good, good. Okay. All right, one last thing. You don't need to write anything on this slide, by the way. Just wanted to show you, this is actually what Mendel was actually able to find from all of his experimentation, from all of his Punnett squares that he put together when it came to his pea plants. He was able to determine that, hey, the dominant trait is more so for round peas than for wrinkled. Same thing with the pea color. Yellow was the more dominant one that was present. Then you have dominant color of flower, and we just walked through the flower part of it and found that, hey, it is a purple flower that is dominant, more dominant than our white. And they have the ratios up there of our phenotypes or our expressed traits. So for every 4 plants, you're going to have 3 purple and 1 white. And obviously there's like decimals in there, but you can't have like a 1/100th of a plant or 1/100th of a pea, so it's going to round to the nearest number. So a lot of these are going to be 3 to 1 ratios. I think all of them are going to be 3 to 1 ratios looking at them. Yep, all 3 to 1 ratios for dominance. So he was able to show that, hey, with monohybrid crossing, you should always have a ratio of 3 to 1. Regardless of what trait you're looking at for monohybrid crossing, you're going to get 3-to-1 ratio in the end. Questions over anything we did for today with the monohybrid crossing?